Okay, welcome everybody. So today I want to show you how we use the surfer for drug discovery for one of our important targets, which is a sodium-dependent amino acid transporter. We use the surfer 96SE for this data, and the goal of this work was, of course, to find inhibitors for this transporter. We have two different target indications, which I cannot really name you at the moment. And of course, for this transporter, and this is pretty often the case, the pharmacological tools are not available. We use for our project a decent screening cascade. So we start with a membrane depolarization assay and do a high throughput screening with this technology. Afterwards, we validate the hits with the amino acid uptake mass spec assay and a cytotox assay in parallel, more or less. And afterwards, we again validate these hits of the both assays we used before with the surfer technology, with an electrophysiological assay. Because in the past we had some other transporter projects where we don't have this assay available, and normally a lot of compounds we identified were not direct transporter inhibitors. And afterwards, of course, we go forward with tertiary assays, with afterwards in vivo testing and so on, which is not described here. So the goal clearly for this task was to develop an electrophysiological direct transporter assay, and actually this should have sufficient throughput to run an SAR campaign for this project. And the plan for us was to do an assay development with the nano surfer technology and try to make this assay technology feasible for this transporter and conduct the compound testing afterwards. Normally, we have some overexpressing cell lines available with this transporter, so we produce a bulk cell culture batch with our automated cell culture platform. We harvest the cells afterwards, we isolate the membranes, and then before the measurements, we prepare the SSM chips with the target, which normally we need 2.5 to 8 hours for that. With a 96-well sensor plate, and afterwards we measure the currents and the inhibition of the currents. We need more or less an hour for a 90-60-well plate. On the right you see the surfer in our lab, which really done a good job for this project till now. On this slide you see a brief assay overview, not really detailed, but just what we're doing during the assay cascade. So we coat first the gold sensor with the solid supported membranes and attach the target membranes. Afterwards we check the quality of the preparation, and then we apply the buffers via the automated perfusion system, and then we run the compound testing with a couple of low controls and high controls on every plate. The assay buffer you can see on the bottom left we use, so it's of course with a lot of sodium inside and with the substrate plus minus, and on the right you will see one exemplified current where you really see we get nice currents after assay buffer and amino acid application. That this current is really driven by the transporter. We did a lot of testing, so we used on the left wild-type membranes plus sodium and plus the substrate, and you see no currents. On the bottom left you see the amino acid transporter currents without sodium and leucine. It's also no currents. On the right, just with sodium you see no currents, and just with leucine you see also no currents. You only see currents if you add sodium and leucine, and this is of course pretty nice with the surfer to see this direct substrate specificity. Afterwards we really measured the Km for leucine, and this is comparable with other techniques. For example, there's a published radioactive flux assay with a Km of 1.2 millimolar, and with the surfer we have around 1.8, which is more or less the same. With the flipper assay and the radioactive uptake assay, it's 1.5 or 1.2 respectively, and this is also everything, so it's more or less comparable related to the Km values. On the next slide, we have really a rubbish tool inhibitor, I must say, which was described that this compound inhibits a lot, but also this transporter. It does not have really a high potency. The IC50 is around 30 micromolars, which is not really good. But nevertheless, we can see in the surfer experiments that this inhibition is, of course, dependent of the concentration of amino acid we use and the time of the incubation. You see with an increase of time and a decrease of amino acid concentration, you see an increase of the block. The IC50 we derive with the surfer is comparable to our mass spec assay, which is of course a good control. Now I want to go a little bit into detail of some statistics of our surfer measurements, because this is of course for screening and profiling campaigns pretty important. With the 96 SE, this is an exemplified plate, so we got a success rate of 98%. So just one or two wells were not functioning, and this is what you can see. This is pretty good. Normally, this is I show on another slide a couple of plates, and you see the success rate over several plates. On the next slide, also with our crappy tool compound, you see really over the plate really good robustness or reproducibility of the block. So this is really something in the middle of the IC50 curve, and the standard deviation is really pretty good. And also on the next slide, I show you the same more or less for a couple of plates. And this is the data I'm referring to. On the left, you see the currents as a signal to noise factor and the low and the high controls, and this is pretty stable. On the right, you see the inhibition of the currents and the success rate. So the success rate is mainly above 80 or 90%. Just one plate was something at 70% due to whatever reason. And you can also see here that the inhibition over several plates is pretty stable. So we were very, very pleased with this assay and start the compound screening with this approach. First of all, we run a trial set of several inhibitors, which are also active in the mass spec assay, and you can see here the correlation is pretty well between the both technologies, which is of course good for the mass spec assay, but also good for the surfer assay. So this also indicates that our screening cascade is pretty good. But nevertheless, with the and this is just show you 5 examples of some compounds with the data in several assays. Nevertheless, we found with the surfer that some compounds which are active in the membrane potential fluorescence-based assay and the mass spec assay were not active in the surfer assay. And after several other experiments, we really can devalidate this compound or this series, which was one of the first series out of the HTS. And nevertheless, after the compound optimization, compounds which are not active in the surfer are devalidated. We don't pursue them any further. So this is really a gatekeeper for in vivo testing. We also try to establish some structural biology approaches, but nevertheless, it's not really easy or it's not possible till now to crystallize this transporter, but nevertheless the bacterial homologue is crystallizable, and so we try to produce a high amount of the bacterial transporter for some crystallization experiments, and also to do some mutant analysis afterwards. But nevertheless, the big goal here is to show that the bacterial transporter we are isolating is still active, and also with the mutation we want to use afterwards are still active. Therefore, we also build up a surfer assay for the bacterial transporter, But this time we reconstitute it, the transporter, into proteoliposomes, which also works pretty nice, and you get higher currents compared to the CHO membranes we use normally. And so this was also validated that we can use a surfer assay as more or less a quality control for this approach and for transporter batches, purified transporter batches. And there are some other mutant analysis and also another bacterial transporter we also want to try for structural biology. We are working at the moment to validate these mutants. We also see that there could be some, or we know from the other project that there are sometimes differences in the block between different species. So we want to also check if the mouse transporter or the compounds are also active on the mouse transporter. Therefore, we also try to develop an assay with the CHO membranes with the mouse transporter inside, but nevertheless with the N96, which was a machine we used before, it was not able to see currents with these preparations. But nevertheless, and this was pretty nice for the 96 SE, because with this machine, which seems to be more sensitive, we see also currents with the same membrane preparations with the mouse transporter. And also the success rate was here pretty high. But what is pretty clear, the currents of the mouse transporter are smaller compared to the human transporter, which you can see here. The Km is more or less in the same range. But we also check with the surfer now the compounds also on the mouse transporter, which is important for further in vivo testing. And last but not least, I want to show you the summary. So we use this the surfer technology for this project as more or less the assay to validate the compounds, because this is the only really direct assay we can use. The assay is robust and reproducible with a high SN and success rate. It's automated, and now the testing of the compounds, really we do not need a lot of time for that, and our throughput is really reasonable. And so we successfully implemented this transporter assay into our screening tree, and the screening tree is therefore, in my opinion, pretty good for this project. The data correlation between the assays for several series was very good, but nevertheless with the surfer we were able to discriminate some unselective or unspecific compounds, which we do not further pursue in the screening tree. So therefore here we have a unique direct electrophysiological assay in hand for this target, and we will further work to optimize the compounds using